Hi, my name is Ceci and I'll be doing the Unit 3 presentation. So this article introduces a concept of the genomic code, suggesting that DNA sequences dictate the 3D organization of chromatin in a human genome. This helps to explain the functional significance of non-coding DNA by showing how it contributes to the genome function and regulation. And so Bernardi, he also highlights the role of isochores, which are large regions of the genome with uniform GC content. They correspond to specific chromatin domains. For instance, we have the GC pore isochore, which corresponds to the lamina associating domains or LADs at the nuclear periphery, and then we also have the GC rich isochores, which correspond to the topologically associating domains, also known as TADs, at the nuclear interior. Overall, Bernardi argues that small DNA sequences influence the nucleosome's spacing and binding, thus guiding the folding and compartmentalization of the human genome. So Bernardi's genomic code presents non-coding DNA as a crucial factor for how chromatin is organized and how genes are regulated. The non-coding DNA mystery revolves around the question of why the genome doesn't code proteins and yet it has been conserved through evolution. For many years, non-coding DNA was referred to as junk or selfish DNA. And so Bernardi proposed a resolution that stated that DNA is not useless. Rather, it encodes structural information that shapes the chromatin structure in the nucleus. This influences the gene expression, gene regulation, and gene function. So in this article, Bernardi proposed a few pieces of evidence. So one being that he identified two major groups of isochores, which were the GC rich regions and the GC poor regions, which constitute of the lamina associating domains and the topologically associating domains. Another piece of evidence that Bernardi proposed that the distribution and composition of short DNA sequences influence the nucleosome spacing and binding, which influences the chromatin folding patterns. And so he also proposed conclusions, one of them being that non-coding DNA plays a key role in shaping the chromatin structure, challenging the notion of junk DNA. He also concludes that the genomic code provides a framework for understanding the genomic organization and emphasizes the functional importance of DNA sequences beyond the protein coding. And so overall, my opinion of this article is that Bernardi allows us to appreciate and understand the powerful role of non-coding DNA. And also Bernardi challenges and expands the views of non-coding DNA, and he also helps to deepen our understanding of how genome architecture plays a role in gene function and gene regulation. That's all. Thank you.